As Gabe said, I'm at University of Chicago. Background is in physics, but over the course of my PhD, I've been studying microbial ecology. And I guess kind of the little vignette slash question that I wanted to pose to you all today has to do with some stuff that I've been working on with Sepakun. And so we're interested in if you were sort of to sample along some landscape. This is a geographic landscape, not a energy or otherwise. And you choose some sort of points in this landscape. And then in each of those points, you measure sort of who is there in terms of the microbiome. So we're talking about 16S data. And sort of what are the contexts? So this could be like soil chemistry information, like pH, carbon, nitrogen, phosphorus, or maybe this is, in the case of like a Terra Oceans type data set, maybe this is like water oxygen levels. Sorry, as you can see, I'm more of a mathematician than a chemist. We want to know kind of like how are these two things relating to each other? So in other words, who is there being shaped by the contexts of where they are? And so in particular, one thing that we've been thinking about with regards to this connection is how does this then relate to sort of phylogenetic signals along a tree? So let's say we have some sort of tree and we can then identify some response of all of these species. So let's say we've measured this in a lot of different locations, and now we're extracting via technical means I won't go into here, sort of primary axis of response and coupled with a primary axis, a driving vector. And so now we have some sort of, I will draw this as these vectors as a, let's say like a bar graph. We now have some sort of response where, let's say these two are with sort of increase along this environmental axis. You have an increase in the abundance. Whereas these two, you end up having a decrease in the abundance. Maybe here you have, again, an increase. And I've been kind of interested in how can we then begin to characterize functional groupings in comparison to perhaps phylogenetic groupings, and where we're interested in like, are things that are closely related, are they responding to the environment in the same way, in comparison to things that are sort of distantly related? So here, like these ones, I said, oh, well, they're closely related, so they're both up. These ones are closely related and they're both down. But then like in this clade, I showed some are up and some are down. So, is it something that's kind of going to be randomly scattered along the tree? Or is it something that is matching clades within the tree and having sort of this coherent response? And so one thing that I've been thinking a lot about is how actually can you quantitatively make an argument about this? So maybe the question that I'm posing is one of more of a technical one to you all, which is I've been thinking a lot about, we see sort of qualitative indications that yes, there is phylogenetic coherence in response. But how does one actually make a quantitative argument along this way? The inclination is, or my sort of intuitive inclination is there some way that I could do like a moving window average along the tree? But what would that mathematically mean? Or, we were trying to do, say, okay, for let's say we take every pair of of ASVs or of species? And then can I look at sort of this difference between the loadings as well as the distance between the two species? And then if I kind of like plot that now along an axis of on the y-axis, it's phylogenetic distance. And on the x-axis, it's sort of distance between response. You know, you can set some like background level, let's say, which is if you were to shuffle this tree. But then what would this curve look like? And what would it indicate if you had coherence or not coherence? So, you know, let's say you had something like this. Well, you know, this would be saying that, okay, things that are closely related are more similar. In other words, they're below the null expectation of distance and response. Things that are sort of in some intermediate region are more different than the null expectation of response. So maybe you have in some intermediate distance, you have like anti-correlated response. And then maybe you have something like convergent evolution again. I don't know. Or you could expect something maybe like this, where you have a difference and then it approaches random. Once you go far enough away, you've completely lost any relation to one. So the curiosity question that I pose to you is if anybody has a better idea of how to actually quantitatively answer this question of phylogenetic coherence, where you have non-binary traits. Because I think there are some really interesting techniques that Martini's lab have done as well, where you have binary traits, and now you're looking at just how far back in the tree can you go maintaining coherence of these binary traits. But once you start to go into continuous traits, how can you address this question of phylogenetic coherence in a sort of convincing and not convoluted, and doesn't require I give you 30 minutes of talk about the technique to explain the result? Yeah, so that's my curiosity of the morning. Yeah.
Yeah, I think this is such a beautiful and fundamentally important question for Earth, because it's about the redundancy of biodiversity and whether biodiversity matters. So my question is, it seems incredibly important what scale you're asking this question on. If you ask for all of life, all three domains, you'll get a very different answer than if you're just looking at bacteria or some subset of bacteria. So what scale do you think is most important for this question?
Yeah, I mean, I guess for practical reasons, I've been thinking about at the bacteria scale.
All of bacteria?
Concentrated to a specific microbiome. So like thinking about just bacteria that are in soil. But I do think that this is like, one thing that I've been interested in is even just like, what if I were to subset and look at only the bacteria in like one continent or one country versus then across somewhere else? And what if you subset between phyla or something like that?
Yeah, sorry, I didn't mean to interrupt. It's interesting, that question, but I really think that you'll get a totally different answer if you look at just the gram-positive, and then maybe within the gram-positive, a genus, or do you know what I mean? Like, I think that'll really shape how you, what answer you give.
Yeah, thank you.
That seems like a feature.
Like
not a bug. Seems like a feature, not a bug.
Yeah.
Yes.
So you're using like different approaches to quantify phylogenetic signal, like Blomberg's K or Hagel's lambda or anything like that?
I have not, because I have never even heard of them, in fact.
Okay.
So
it's like an approach that basically kind of assumes Brownian motion evolution along that phylogeny, the idea being that things that are more closely related have similar kind of traits, and it's especially for quantitative variables like yours. And so it kind of varies on a scale of like 0 to 1, which means that, these things are kind of closely related and showing similar trends. So there's some, cool like phylogenetic comparative methods that kind of get at that. Or like, I was wondering if like some sort of a Mantel test or something would work for like the figure you have on the right there, where you have like phylogenetic distance and like a dissimilarity matrix of responses or something like that.
Those are all really cool ideas. I will follow up with you so I can write them down when I have a
Cool.
paper in front of me.
Cool.
Really quick, Milena. So you posed this problem to us at the beginning rather vectorially, right? You said there are many microbes that could be here. They enjoy some like distance measure on some phylogenetic tree, and yet the sort of stimulus that they could be responding to are like, like there are many things that the environment in environment. So I'm wondering how you're thinking about this from the like conditioning on like all the many possible sort of axes, not just the scalar that you put on the y-axis, but the sort of like spectra of variation functionally that you may be thinking about.
Yeah, I mean, I guess I sort of have been thinking about them as somewhat separate problems in the sense of the first one is how do you extract the relevant axes within environment, sort of conditioned on the assumption that in fact these microbiome are responding to the environment in the first place. But sort of how do you extract those relevant driving vectors, and then can we sort of find corresponding response vectors as kind of one problem, and then the secondary problem being how do we interpret those response vectors when you're talking about tens of thousands of sort of amplicon sequence variants or roughly sort of independent strains. So I've been kind of thinking about them as separate problems, but then looking at sort of characteristics or like what comparing across then different directions and seeing like, do you have different types of coherence in these response vectors depending on what environmental direction is being coupled to. I don't know if that answers your question. So not so much.
You find dominant direction.
Yeah,
you do
exactly.
the sort of a dimensionality direction in the variation and environment phase. And then you ask along these axes, what are the correlates? Okay. Wonderful.
Sure.
Thank you.
Yeah, thank you again.